The next group of Enterobacteriaceae that we'll talk about are the Shigella. Bacterial nomenclature and bacterial phylogenetics are complicated because new genetics and new molecular tools really cause a lot of rearrangements in how we think things are organized. And then it gets confusing because we've spent so long talking about bacteria one way and technically it's wrong. So I'll just point out the technicality that Shigella technically is not its own genus. It's technically a subgroup of E. coli, but that's confusing. So we're still going to call them Shigella, and we're going to pretend they're their own genus. So I'm sorry that that's confusing, but that's just the way it is. And maybe someday they will reclassify them officially, but this is just the way we do. Just like with Salmonella, the Shigella, I'm going to call them species, that's fine. Shigella species can attach to M cells. They do have a type 3 secretion system, and they use that to induce phagocytosis. They actually want to be phagocytized. So they induce their own phagocytosis. Once inside, they will lyse the phagosome and replicate in the cytoplasm. They can then pass cell to cell. What other species did we talk about that can move from cell to cell without exiting the cell? That's a really cool immune evasion strategy. Shigella dysenteriae also produces Shiga toxin, just like the Shiga toxin producing E. coli. The Shiga toxin, like I mentioned when I mentioned it with E. coli, causes cell death through disruption of protein synthesis. So it's one of those AB toxins that the binding domain binds to host cells, the active domain gets into the cell, disrupts protein synthesis, and leads to cell death. For Shigella, humans are the only known reservoir, and this is different from E. coli and Salmonella, where those are found in lots and lots of animals too. So it is not yet known to find Shigella inside of other animals. It is transmitted through the fecal-oral route, as you might expect. This one is dangerous, just like the S-Tech, where it's very few cells needed for disease, from 100 to 200. It's estimated 500,000 cases per year, 90 million worldwide. Shigella sonnei accounts for about 85% of human infections. Dysentery is the more dangerous one, but sonnei is most common. Shigella flexneri is more common in developing countries. And then we do see outbreaks of Shigella dysenteriae periodically. I did mention it's more dangerous. It has a 5 to 15% mortality rate. The other Shigella infections, just like with most GI infections, will be self-limited. Some days of diarrhea, abdominal cramping, not fun, but generally self-limiting, whereas the dysenteriae can be more dangerous. With Shigella, you're very commonly going to have cramping, diarrhea, fever, and blood. Okay, so you're going to have bloody stool because of the tissue destruction. The first signs of disease are caused by enterotoxins, and you're going to also have, this is kind of unique, pus in the stool. And that's because of the invasion in the colon. So you're going to have, white blood cells coming in, you're going to have the bacterial cells, so you're going to have pus in the stool, which is not generally what we see with E. coli or Salmonella. And so I know it's unpleasant to talk about, but sometimes just looking at a stool sample can be maybe not 100% diagnostic, but can really help narrow down the potential cause of infection. So if you have a patient that shows up with pus in their stool, it's maybe Shigella, and that's something you can think about. As I mentioned, with most GI infections, this is usually self-limiting. Some individuals can maintain carriage. We are starting to see levels of drug-resistant Shigella in the U.S. Not up to 100,000 infections yet, 77,000 infections per year with fewer than 5 deaths, but still, in patients where they're having trouble clearing the infection, antibiotics would be prescribed, and if it's an antibiotic-resistant strain, that can be very dangerous. We'll leave poop for a little bit, and we'll move on to another genus, Yersinia. Even though this bacterium is part of the Enterobacteriaceae, which makes you think of gastrointestinal infections, Yersinia pestis actually causes the plague. Within the genus, there are species that can cause gastrointestinal infections, including Yersinia enterocolytica and Yersinia pseudotuberculosis, but plague is the one that everybody likes to talk about. As with many members of the Enterobacteriaceae, they are resistant to phagocytic killing. They have that type 3 secretion system that helps them to induce cytotoxicity in phagocytic cells. They can induce apoptosis of phagocytic cells. They can suppress cytokine production. So they have a number of different mechanisms to help them evade the immune system. Yersinia pestis has 2 plasmids that contain virulence genes. The first plasmid has a gene that encodes proteins for anti-phagocytic capsule, so that helps them to evade the immune system. The other plasmid has genes that help them to degrade C3b and C5a. I didn't ask you to memorize the complement cascade, and I'm still not going to ask you to memorize the complement cascade, but these are proteins that are important for complement. So basically this helps them to evade complement, and it also allows them to degrade fibrin clots so they can continue to invade the body. Yersinia infections are typically zoonotic, especially when we're talking about pestis, which means that they're supposed to be found in an animal life cycle. Humans are an accidental host. Different species of Yersinia have different natural hosts, but again, Yersinia pestis has caused massive pandemics throughout history. And when we talk about the life cycle of Yersinia pestis, this is an area where a little bit of knowledge of ecology is helpful, which is why we make you take ecology. Plague can be maintained in an urban or a sylvatic cycle. And so with the sylvatic cycle, that's like the natural cycle where you generally have fleas and things like ground squirrels, other rodents that live in like the forest. Humans can accidentally encounter that when they're camping or hiking. There have been cases of plague out of Yosemite and the Sierra. With the urban cycle, you're generally looking at these non-native rats and their associated fleas. So it's relatively easy to control urban plague because you just control the rodents. So in large cities that have really good rodent control, you don't see plague, but we've had plague in urban cycles in Los Angeles in the early 1900s prior to better rodent control. The major Black Death that people talk about, in the mid 1300s, you can see the spread of plague as it came from Asia. So on a couple of the major trade routes, and then the orange, they started to document plague in 1347. So again, these major land trade routes, and then the ports. So Naples, Genoa, Marseille, and then it started to spread from those ports, 1348 into, other parts of Italy, Paris, London, other parts of Spain, 1349 into Portugal, Northern Africa, kind of Eastern Europe, and then spread. So it was one of those that, because of how slow travel was at the time, it took years to spread. In modern times, this would have spread very quickly. There are two manifestations of plague that we talk about. There's bubonic plague, which is like the Black Death that people are familiar with, and there's pneumonic plague, which is actually more dangerous. So the bubonic symptoms, it's called that because you get the formation of what are called buboes, or these swollen lymph nodes. And these lymph nodes will contain, you see this under the arm, this really large swollen lymph node where the bacteria are just replicating like crazy. This can go septicemic, and then when that happens, the mortality rate is like 75%. What happens here, and why they call it the Black Death, is because when the bacteria go septicemic, you get necrosis, especially starting where you have capillaries and lower, like thinner blood vessels that are more likely to get blocked by the bacteria. So you start to get necrosis of things like the fingers, and that can spread. Pneumonic symptoms will develop far more quickly, within 2 to 3 days. Fever, tiredness, upper respiratory symptoms. This is highly, highly infectious, person-to-person spread. And it has approximately 90% mortality. So the bubonic is acquired after being bitten by a flea, usually. This is not commonly person-to-person. The pneumonic form is. So in the US, we do have plague. This is the US map from 1970 to 2020. There's a dot placed in each county where the plague was reported. What maybe you'll notice is that it tends to be found west of the Rockies, so California, common in the Four Corners region. This case right here in Chicago was a laboratory-associated case. And then since 2000, there have been at least one case every year, a handful of deaths. 2015, there were a lot of cases, 16 cases, 4 deaths. Even 2020, 7 cases, 1 death. And then worldwide, there's a lot of plague in some parts of sub-Saharan Africa, Madagascar, then South America and the US. So plague is not one of those infections in the US that's common, but it's also not unlikely. So plague killed a cat in southwest Idaho, sickened a child. What is the plague? Why is it here? These are the types of diseases that people hear about and they're like, what? That's a disease of like ancient times. But it's really not. We still have it. This is from 2015. Second case of plague, another Yosemite visitor investigated in California. So we do have them. Camping is canceled at Phish's Colorado concert. Blame the plague-infected prairie dogs. So plague is common in animals, very common in animals. And every once in a while, it can jump into us too. Other Yersinia species that can cause gastrointestinal infection, you have Yersinia enterocolitis, right? Gastrointestinal infection. From contaminated food and water, 4 to 6 day average incubation period, diarrhea, fever, abdominal pain. It can mimic appendicitis. So you'd want to rule out appendicitis in a patient that shows up with these symptoms, because appendicitis can be fatal far more quickly than a Yersinia enterocolitica infection. It can be a chronic infection, but it generally will respond to antibiotics. Yersinia pseudotuberculosis will cause very similar symptoms to enterocolitica. However, it can be a little bit more invasive, so you may have patients that would show up with things like septicemia, abscesses, liver, bone, and muscle infections.